You're full. Well, you're in the corner now. That's yeah.
Hey.
Tiny milk.
It's coffee.
With milk or without?
With coconut oil. Coconut oil. Yeah, with coffee.
Fancy?
It actually has a name. Called, let me check.
Voordat je alles opgesteld hebt, natuurlijk. Sorry. Heel erg vervelend.
It's called a waterproof. Waterproof. Waterproof coffee.
Ah.
Ja.
I heard that it's very
Ja, maar ik weet niet of het heel handig is om hier te zitten, want dan zit je dus op de kamer.
maybe that area. It's very pleasant. It's so strong. How is your weekend? Just looking on instead of
Nee, helemaal nog. Oh, lekker.
Today
Okay.
before we get started, how do you like the course so far? Awesome. Besides awesome, which is what we build, who feels a bit frustrated? Oh, you are overwhelmed. Overwhelmed? Yes? Okay, that's better to be a bit overwhelmed than to be frustrated. So who feels very frustrated? Well, if you don't want to show your hand, come and see me or anybody else, right? A bit of overwhelm is quite normal in the first few weeks because everything happens at the same time. Everything is new, right? So that should get better during this week, hopefully, not next week. But if you still feel overwhelmed by the end of next week, also come or before if you want to. But certainly by the end of next week, come and talk to one of us. Yeah? Because it's okay if you feel overwhelmed at the start, but you shouldn't stay that way. Right. Who recognizes the spot? Maybe someone who recognizes the event. Yeah. Because this was I actually don't know when it was exactly. Does anybody remember which year it was? 2 years ago? Something like that.
I just have one question.
Yeah.
The readings, the lectures are now being put up quite late.
Oh.
And in the schedule is not like reading until
Oh, it shouldn't be.
yeah.
Yeah. Okay. I'm not amazing to do this. Can somebody remind me to take a snapshot of the to-do list? Oh, I'll put it here, then it's in the video. Chalk. Can you stop my chalk?
I think it's might be there.
There's a little box over there.
Oh, yeah. Thanks. It's always much easier to spot things from up there. Read it. Right. Any other practical things that you've run into that you would hope that we can solve? The question was, can you put up the reading material earlier? And also we put up the detailed schedule on Canvas, the one that I showed you in the first lecture. But apparently the reading column went missing because it's empty. It shouldn't be. I don't know why. Yeah, but so the question here is, wait, first, can everybody hear me well enough in the back? Yeah? Just, it's not, I'm not talking too loud, I'm not disturbing you. Well, if you start missing me, just wave, and I'll speak up. Because I like this much better than standing with a microphone. Because it gives me the feeling that I actually have to do work. And that it is. Okay. So your question was the PowerPoint, the lectures. Well, usually we're doing final touches just before the lecture, like in the train on the way here. And it's easier for me to just put the final version on. But we also have last year's lectures. All of them are already on Canvas. You have to scroll down a bit. So there's a module that says Lectures 2017, if you haven't found it yet. And the lectures are usually quite stable. Yeah? So you can review those, and then there may be like a few slides if they want to be yeah? So that we show for you. Yeah?
Also the tutorials for the like informatic class, can you
For the programming?
Yeah, because they're always like just after the break, but, like if you're already there and you just want to see some program, can you even like update it earlier, or because it's always
The tutorials.
Yeah.
To put them up earlier as well.
yeah.
Yeah, we'll try, but we're actually updating those for the very practical reason that we have they were actually designed when there were 20 students in this class, in this course. And that means that it involves a lot of interaction with the assistants, which is very difficult for us to keep up now that there are so many. So we're redesigning some of those tutorials. So
Okay.
but I'll try.
Like you can already see what you need to do or
Yeah,
something? Or
I'll
because it feels already a bit late.
Yeah.
And,
Okay. Any other practical things? Yeah?
When you do go to the programming class,
Yes?
you come across really silly things like a room is missing almost all its chairs. It's really messy and it creates kind of like chaos and problems for them.
Yeah. So you guys just fit in the rooms we have. So that also means it only fits in one way, and it only fits if all the chairs are there. We can't control whether other students, classes of students, have been borrowing the chairs from the rooms that we use. Like we also do sometimes when there's more people in the room than we have chairs available. So that's hard for us to fix. The suggestions that Adele put on Canvas yesterday, to go sit in the same room that you were yesterday, that should work. If everybody does that, then it fit yesterday, it should fit today. Right? And I think that's the only thing we can do there. And it might be that you have to go back, well, not from Monday on Tuesday, because I don't think these rooms are used in between by others. But from Tuesday to the next Monday, then likely some of those chairs have gone written. So yeah, we just have to yeah. So in the structural bioinformatics course, we'll go into entropy, and remember this example, right? Diffusion and entropy. There's nothing physically difficult with fitting all of you guys in the room. The problem is that there's only one configuration that works well. That's entropic cost. Yeah.
Will there then also be TAs in all the rooms? If there's a day we stick to our room and then someone can in, oh, there will be TA in the room. That's hard.
That was for the programming class.
Yeah.
So for the programming class, we use all the. You're getting ahead of me. So for the project, we need 4 rooms. So we have the 3 on the 3rd floor and 1 on the 4th floor. For the programming, we want to use only the 3 on the 3rd floor. Yeah. Okay. Right. So other practical issues. Now we're at it. No. Okay. That's good. If there are any, you can email me or you can ask any of us during, me during the breaks or any of us during classes. This was, to come back to the opening, this was 3 years ago, I've been told, and it's actually at the FoB. It's actually, Dorian, get my bearings here. I'm not, it was just over there, where one of the main water pipes broke. Just below there, you can see the cavity. There was a parked car here, actually. And I think this one disappeared later in the day.
Your videos have been floating away.
Yeah. So this, I was there. Everybody was there to have a look. And they actually flooded the basement of the manufacturing building, and for reasons that are probably always going to stay mysterious, all the IT equipment is in the basement. Probably because nobody wants to have an office there. And so that was quite a disaster, to put it mildly. Okay. That was, so that's what happens if you have a flood of stuff that you don't control. It goes everywhere except where you want it. Yeah? So having a lot of water is nice, but if you don't contain it in the pipes where you want it, you're going to have a problem. The same is about data, right? And you're the guys and girls. Can I use guys in a non-sexist manner. Yes. If you're offended by that, just come talk to me after. You're the guys that we're trying to teach you how to build the pipes containing the data, yeah? And even to do something useful with it. Okay, so sometimes you need a lot, sometimes you need a little. You need just this particular bit, how to find it. That's what data is about. That's what most of our courses are about. So we'll get to that in a minute. I have some more practical announcements. You already saw a few of them. The detailed schedule is on Canvas, as I already said. We'll have to look at the missing column, or the column with the missing reading information. You all need to register to the course. Oh, if you're in our if you're in our master program, yes. You have to register to the biosystem data analysis course. Unfortunately, has anybody been successful in that?
Yeah.
Okay. Who has tried but failed? All right. Who hasn't tried yet? Okay, it's about 30. We're looking into how to solve the technical issues there. If you haven't tried and you don't know how, ask one of the other people, because most of you know how to do it. Yeah? So who hasn't tried and doesn't know what to do? Okay. Go find some of the other people who've tried. Yes. We'll let you know when we know the solution to the technical issue. But maybe sometimes it just works if you try again. As you have already noticed, because otherwise you wouldn't be here, because we had to switch rooms at the last minute, where I think we're not twice in the same lecture room. Maybe a few times, but we're usually in a different lecture room. Yes?
How do you know for sure that you're enrolled for the course in UVA?
That's a very good question. I have no idea, because I was never a student here. So I don't know how the system
I
works.
think you should get an email.
You get an email with information?
Yeah.
Yes? Okay.
You get an email and you can check it in the roster in the UVA site.
Yes, okay, so if you check the UVA schedule site, it should show up. If it doesn't, you're not enrolled. But ask one of the TAs, because they've actually been a student and they know how they did it, or at least they probably remember how they did it last year. Lectures are in different rooms. Right, this is what I already said. So for the project, we have all the 4 rooms. Please go back to the room that you were in yesterday, then it should fit. For the programming, we use the 3 rooms on the 3rd floor. Very easy to remember, 3 rooms on the 3rd floor. It's easier for us to be able to shift assistants to another room when there's a lot of questions here, but fewer questions there, and then shift back again when it's reversed later. Yeah. And I actually also, you might have noticed that I'm going from room to room. Okay. Some of you guys, most of you guys have signed up to their project group. Some of you haven't. Who hasn't signed up to their project group on Canvas yet? Right? If you don't know what I'm talking about, ask the other people in your group. If none of you know, ask another group. Yeah? Please sign up, because then we can keep track of who is in what group, and you'll actually get the grade. If you're not signed up in your group on Canvas, you don't get the grade for your project, you can't finish the course. Oh yes, because there's so many of you and we'd like to get to know you guys, it's easier if we can match names with faces. And like right now I can see all your faces, but I don't know your names, and when I'm looking in Canvas I see all your names, but no faces. So please add a profile photo in Canvas. Who already did that? A few of you did. Oh, most of you did. Okay. For all the rest, please do it. If you don't know how, ask one of the people who managed to do it.
It doesn't show on my profile, for example. If you've uploaded it, but it sometimes doesn't show. But
I don't know.
maybe on the CNET.
Yeah. I don't know. Okay. Check your Vue email regularly, or if that doesn't work for you for whatever reason, set up forwarding so it goes to your personal email address. Because you will get announcements from us, you might have already seen them, I hope you have. but if you miss them, you might have issues at some point.
I was still not able to forward my
I don't know how I don't know how to do that exactly. So for practical issues, please ask the TAs. They usually know. Right, and then oh yes, which group hasn't changed FVM password yet? Really? All of you? That's good. I'll check with boss if that's correct. Right, now back to the data, to the lecture. So, if I say life science data, what do you think about?
gene expression.
Gene expression. Anything else?
Protein structure.
Protein structures. Anything else? I can go on like this for weeks because there's so many. Right? Sequencing data in general, yeah. It could be expression, could be genome sequences or copy-number variations. It can be imaging. It can be proteomics. It can be post-translational modification. Well, that's proteomics as well. It can be, well, what else do we have? Anything else? Oh, the metabolomics, yes, very good. Hmm? Oh, right. Yeah, so cancer epitopes. Anything else?
Transcriptomy.
Hmm?
Transcriptomy.
Transcriptomy, yeah, that's expression. Yeah, so, yeah. Okay, so. Let's see, let's step back. So we have two use cases from one is both of them are actually from our research, so let's say you have transcriptomics data, so RNA-seq from bacteria in your mouth. So you take a mouth swab, you sequence all the RNA in it. A lot of that is your own DNA, but all the rest is bacterial or viral, and then you want to compare a healthy mouth with a diseased mouth, so caries or gum disease or something like that. And you want to know which pathways might be deregulated. What data do you need? Well, obviously the RNA-seq, you have that. But which other data do you need?
Clinical data.
What kind of clinical data?
Disease symptoms.
All right, so you need to know who's healthy and who's diseased, yeah? Or maybe even what specific type they have. So write it down. What else? You need a lot more. The database, you can refer what database of what? With RNA-seq. But what should be in the database? Sorry? Strains and pathways, right? Okay, so the database of strains or species. The database of pathways. Can you hear me when I'm speaking in the board? Yeah, also in the back. Anything else?
You need a database in which RNA gives which protein.
Right. So, gene annotations. Yeah? What else? Reference sequence for what, of what?
Yeah, it's in the database, strain species.
Well, you can say reference genomes, right? Because it's not necessarily quite the same thing. So this is a relatively simple example, and you need already at least 5 types of other data. Now let's go to the other one. So you're looking at mutation data of the tumor sample, right? And then you want to filter out those mutations that are likely associated with the well, the occurrence or even the forming of the cancer. We look for a different word. The development. Yes. So what data do you need?
Data of
Okay, so known tumor genes. We need more than that. Yeah, yeah. So that's what they call it a matched normal. So you want to have because if you have sequence data of the tumor, then you don't know which part of the sequence is a mutation. You need to have the normal DNA of the same person as a reference. Right? If you don't have that, because sometimes they are not available, then you can just take a reference genome. Or what's also done, if you have like a library of data from tumor samples, you take the consensus sequence of all of them on the assumption that most tumors will be unique, so the mutations will be in slightly different places, so the consensus will be the normal human sequence. Yeah, with some deviations. It's not perfect. This is better. Okay, anything else?
You would also want a gene annotation.
Gene annotations, exactly. Okay, I think you get the picture, right? So to be able to do something with data, you actually need more data. Yeah, and that's also part of what makes bioinformatics so interesting, because it's about figuring out how to connect all those different types of data in an efficient way to be able to answer important questions. Yeah. Okay. So we'll go a little bit into the details of these two examples. So we have two cases for the oral cavity. We have swabs from healthy and swabs from diseased mouths. We have the DNA. We do some data processing. Yes, and then we get different pathways. Right? We can say, okay, this pathway maybe for fatty acid metabolism is upregulated in the diseased case, and some other pathway is downregulated. And here you're looking at sequencing data. So here you have your reference genome. Do we have reference genome? No, reference genome. You line all the reads, you look at differences, and you pull them, and then you annotate them on, again, a reference genome with gene annotations and known tumor genes, maybe also gene functions and so on, right? Because the next thing you want to know when you see a cluster of mutations on a particular gene, you want to know, what does this gene do? Do we know anything about it? Is it already implicated in cancer? Or is it implicated in some function that we can associate with the development of cancer? Yeah? Okay. Oh, interesting. We already had this slide, didn't we? So, yes. So we already did this. Right. So to put it a bit more general, so there's a lot of public data that you can use. So you could mine literature to find data for comparison. You need data when you create models, and particularly for bioinformatics, if you're building methods, whether they're analysis methods or prediction methods, you need a benchmark, and benchmark is always data. Right? That's what the project is about. It's benchmarking homology detection, right? And you need data to do that. We'll get into later in this lecture. We'll get into the three databases that you're working on in the project. Okay. And the main issue with having the data is your statistical power. To be able to say whether a gene is really different in a diseased case versus normal, you need enough samples. To be able to say whether your model is a good fit with the data, you need enough data to be able to say that's significant. The same for the benchmarks. If you don't have enough data for different cases, with multiple testing, who doesn't know what multiple testing correction is? Okay. So you know what p-values are. Right, okay. So let's say you detect something. Your prediction method predicts something with a p-value of 0.05. Right? Or let's say it's 0.03. Right? So it's just below your normal threshold for significance is 0.05, which means you still have a 1 in 20 chance. This is not strictly correct the way I say it, but for brevity, which translates roughly to a 1 in 20 chance that your prediction is still incorrect. Yeah, so that's good enough for most cases. I wouldn't use it for safely crossing the streets, for example. Yeah, because if you do that on a daily basis, then I think you might be crossing the street 20 times a day. Right? Then you end up having a fairly high chance of getting hit by a car on any given average day. So by the end of the week, you are very lucky if you've survived the week. That is multiple testing correction. Right? So although you can significantly safely cross the street once, if you do this 100 times with a p of 0.03, then you're probably going to have 3 cases, roughly. Did I say it correctly? Yes. Where you're wrong, yeah. And then it depends on your task how bad it is to be wrong. But if it's crossing the street, yeah, you want to be safer than that. If it's diagnosing cancer, what if you're fairly certain that you don't tell somebody they have cancer when they don't, but you want to be even more sure that you don't miss them when they need treatment? Yeah, because if you say to somebody you definitely don't have cancer and you're wrong, that's possibly even worse. Yeah? And then there's all sorts of trade-off, because treatment is also not how to say that. Well, let's say it's not easy. Yeah? Okay, so you also don't want to treat people when they maybe don't need it. So there's lots of balances there. But okay, right. So if you're selecting significantly different genes, yeah, expression or mutations or whatever, there's roughly 20,000 genes in the human genome. Right? So if you take a p-value of 0.05, for any test you'll find hundreds or maybe thousands of genes that pass your test, that you predict to be different, but it's just random. Yeah? You know that you're going to end up with so many false predictions if you select for a p-value of 0.05. So you have to do this multiple testing. So that means you need a lot more data than you might think, because for one gene maybe you can get away with 10 or 20 or 30 samples. But for 20,000 genes, you need to do something smart. There's lots of tricks around this. Not all of them involve more data, but it's always easier if you have more data. Okay, you have to have data of the right kind, though. Just more data doesn't help. Yeah? That's the hardest part. Okay. Good. Now, what are we going to do in this lecture? I'll go through a bit of sort of an overview of the different types of databases, primary data versus derived data. So which of these would be primary data? Clinical data, I think so, yes? Anything else? Reference genome, I think so? Depends a little bit what you look at it. The other stuff? Gene annotations could be, but it doesn't have to be. If somebody's actually gone out and measured what the protein does that's associated with this gene, then that's primary data. But if if you have a closed homolog, and you say, oh well, it's probably going to be the same function, then it's derived. Yeah? Okay. And then, yes, and machine readable versus human readable. It's very nice if you have blast results that you can scroll through and see if there's genes in there that you like, maybe to use as a template for homology modeling, to make a structural model of your structure, or to get a first idea of what the function of your protein of unknown function might have, because if you have like 10 hits and they're all alcohol dehydrogenases, then it's likely, or annotated as being alcohol dehydrogenase, then your protein is probably also a dehydrogenase. Right? But you can't do that for 20,000 genes. Right? So in bioinformatics, we don't want to have this human readable stuff. It's nice to check, but if it's not machine readable, we can't use it, not efficiently. This is where the big bottleneck is as well. Having the right kind of data and having it available for easy automatic reuse. Those are the current main issues in bioinformatics. We'll come back to that a few times. Oh, it's already here. That's called interoperability. Your script can read that database and then connect the data without making too many errors. I'll go through a bit of an overview of where how to find new databases. We'll go into Go, Scott, and PFAM. All the reason for that should be obvious to all of you. And I'll end with some of the overviews. So, one way to look at data in life sciences is the classical central picture of molecular biology. Is along the axis of genes. What comes next? You've seen this slide probably in the art lecture last week as well. Right? Genes, RNA, right? Proteins, and then metabolites. Right? So there's data associated with the genes, with the sequences, and so on. Also things about the genes like epigenetics, methylation, chromatin packing, other accessibilities, 3D organization in the nucleus, and probably 10 or if not 10, then 100 other things you can associate with the genome. Then there's transcriptomics, with also lots of different flavors and types of data that you can have. There's a lot about proteins, structures, presence, alternative splicing, post-translational modifications, cellular location, excretion, other things. And metabolites, also with a lot of data, similar to proteins, the presence, the location, can't think of other things. And these things are all related, of course. So just to keep in mind that we're not after the data or the methods per se. What we want to figure out is how these things are related in the end, because that tells us a bit about how life works. So all the applications, let's say solving cancer or finding a better method to get a better treatment for cancer patients, that's very important. But at the same time, we're trying to get a better understanding of how life works. And there's a lot of data that we can use to do that as well. Okay, and for all of these levels and a lot of these different things, we have measurement methods, experimental ones. One of the main reasons that bioinformatics is becoming so popular is because getting this data is getting cheaper and cheaper experimentally, right? And that it's for all these levels. There's also prediction methods and annotation systems for all of these levels as well. I can't go into all of that in one lecture. Okay. It's the first time I do this lecture. The slides are by Sonnet, so I sometimes continue with what the story is. All right, so what I just mentioned here is mainly the primary data, right? So then on top of that, so here, sequences, proteins, structures, and so on. On top of that, one of the most obvious derived kind of data is when you start clustering, right? So similar genes by homology. By the way, what is homology? Many of you got this wrong on the biology questions. Yes? Yeah? There's only one definition of homology. Common ancestor. It has nothing to do with similarity, right? That's how, if we're lucky, we can still detect that two things might have derived from a common ancestor. So we use similarity to find it, but it isn't similarity. Yeah? So homologous proteins. Yeah. So here you can say similar proteins because you need to be able to detect similarity to call them homologous. That's not the same. Yeah. Okay. So two proteins can actually be homologous and not similar, because we'll never know about that. Yeah? Unless we find some similarity somewhere. But we'll go into this in other courses as well. Maybe later this year. No, you'll get into this in the project. Okay. Clusters, you can cluster by sequence, you can cluster by structure, you can cluster by function, and so on. Okay, so so that was about primary, and here it's called experimental primary data. Primary data is always experimental, yeah, in the sense that if it's not measured, it's not primary data. And this is about human readable, yeah. So Wikipedia or blast reports that you scroll through, or observations, or looking at protein structures and so on. As I said, for us it becomes, this is not useless. This is certainly very useful, but for us it really becomes only useful when it's machine readable. Yeah? Or well. There's always ways to make it machine readable, to make a machine that can read it. Yeah, you can write a script that mines text structures out of a Wikipedia article, but it's always a stopgap measure. It's always a MacGyver duct tape. Yeah? You'd rather have all the observations and the data and the logic that went into writing the Wikipedia article represented in some other way that is more close to the reality that you're trying to describe, because that's one of the things. One of the things to realize about data, it's never the reality, even if it's experimental data. It is not the reality. It's as close as we can get. It's always a representation of the reality. A Wikipedia article about the citric acid cycle might sound real. It is real in a sense. It's real knowledge, but it's not the reality. Yeah? That's more complicated. We can't deal with reality. We always have to have a model, an abstraction, because otherwise we can't think about it. Yeah. Okay. So before I get really philosophical, let's get. So you can represent the citric acid cycle as a text. You can represent it as a set of protein-protein interactions. You can represent it as a small bit of metabolic network. Yeah. You can represent it as taxonomy, right? Which proteins, enzymes perform which function where in this pathway. Okay. Are we going? Yes. So, and with this idea of data being representation of reality, you can also understand why humans are important still. So what happens in all the databases is curation. Curation always means a human, a well-educated person looking at the data and deciding what should be in and what shouldn't be, or how it should be in. Yeah? And the assumption is always still that as humans, because we have a lot of context knowledge in our minds, we're better at this than machines are. So that's still true. In 10 years, it might be different. Yeah? So some of you guys might be the ones that design the next level, the next generation AI system that does curation much better than the humans do it right now. At this moment, we're not there. So if you're looking at the database, the citric acid cycle in I think this is KEG, yes. Then a lot of these things are human curated. So that means ideally somebody has gone back to a couple of papers where it's described how the pyruvate dehydrogenase